walkthroughs in a compelling and understandable way, applying what we know about how visual information is going to be consumed and processed by our viewers. I hope that after this talk, all of you will have some new tools and ideas to use in your next visual project. I created this little potpourri that kind of shows the scope of our production. But I would like to start by introducing the science graphics team, which I lead. We're a group of 7 scientific illustrators and graphic designers, which are responsible for creating a large array of visuals. Illustrations for our covers, section openings, graphics and data visualizations for the new section, animations for social and video, and figures for our research sections and online journals, to name some. The graphics team was formed in 2015, and since then, we have made a conscious effort to establish a consistent look and style across every visual element that we create in our shop. Our team is part of the larger visual group that also include design, photo, and web content. The graphics team production is a deeply collaborative effort within the group. All of the visuals we create at some point are touched, edited, or perfected by somebody else belonging to our team. This close collaboration is paramount when you're tied to strict publishing deadlines and large outputs of visuals that the magazine and site demand. The bulk of our production is destined to the AAAS flagship weekly journal Science. But we also are responsible for creating visuals for all our online journals: immunology, signaling, translational medicine, and robotics. Alice Kitterman, who is going to talk a little bit later, has been in the front of this segment of our production. She will be talking a bit about all their nuts and bolts in her work. Our team has to cover the needs at 2 levels of coverage, and also at some spaces in between. On one side of this bar, there are visuals we create for a very special audience of the publication, related to papers and research, with summaries and reviews. And on the other side is the coverage more targeted to a general public on science news. We create and edit a large number of scientific figures, which normally contain graphics, charts, diagrams, with the objective of displaying data and information linked to a very specific paper. These visuals are very close to the reference material provided by the researchers. For example, the figure on the left by Natalie Carey closely respects the content and general design by the author's figure, but has been styled to make a consistent look. The figure on the left is another example by Val Altonian, showing molecular structures generated in 3D software with the data provided by the author. Some of these figures are hybrid. In our perspective section, we use graphics that live in between these 2 worlds, these 2 sides of the audience bar. These visuals are still targeted to a specialized audience in terms of content and topic, but have the expectation of reaching across disciplines. Those graphics require lots of feedback from the authors and the expertise of our scientific illustrators interpreting correctly this information. The mantra or the general idea is that if we create a figure for a biology paper, a physicist could easily follow it. Here you can see an example also about Barolini. On the right, we can see the finished graphic, and on the left, the reference material that she used to create it with all her annotations. As I said, in these pieces, there's a lot of back and forward between these two elements. And finally, on the other extreme of our bar, we have information graphics. We produce them for the new section of the magazine. And these visuals are targeted toward a more general audience. And because of that, have normally a lot more visibility. Information graphics use tools like illustrations, charts, pictograms, diagrams, and animations in combination with text to describe information about current scientific development. Information graphics have the same expectations of accuracy and usefulness than other visuals we produce, but are somewhat more open to the illustration to craft this narrative and flow to convey information. In this example by Chris Bickle, the Hiawatha crater in Greenland, having cut a hole to show its internal structure, giving a sense of the thickness of the ice that covers it. Secondary diagrams explain how the crater is explored with radar from the air. And also, the viewer get a very good sense of the relative size to other impact craters on its location. I'm sorry. Related to information graphics are data visualizations. A good portion of our graphics contains some form of data visualization in the form of charts and cartography. But in some cases, there's a crossover of categories, like this cover illustration created by Barolini. In this visual, the illustrator used some 9,000 points of data from the MAVEN probe to generate the trajectories of charged particles from the solar wind hitting the weak magnetosphere of Mars. Val, who is not a coder, by the way, learned enough Python to code this visualization on Cinema 4D. At this point, you may have noticed that all our visuals, even the ones that follow closely the reference material provided by the authors, have a common feel and flow of information. The reason is that we try to follow certain basic principles. Principles rooted in the world of design and the understanding of how our audience consume visual information. Take, for example, this data visualization piece by Jia Yu and Darwin Gurion. The graphic uses the change in size of geometric shapes, circles, to represent the number of cases in several diseases since 1949. And color, orange, to highlight the moment in time in which specific vaccines were introduced. We can clearly appreciate the effect of vaccination in the decreasing numbers started on those choke points. But in some cases, the abstract representation of information is not adequate to relate the information we're targeting with our piece. We need tools like illustrations, diagrams, pictograms, etc. This beautiful explanatory graphic by Chris Bickle explores the plans to restore Notre Dame Cathedral after the 2019 fire. The graphics show the interior of the building, the exterior. It gives us a very good sense of scale, thickness of the walls, and a whole set of secondary diagrams provide the reader with a vast amount of detailed information regarding how the fire might be affected certain areas and structure. So the question is what those two visuals have in common. One is an abstract statistical piece, and the other an explanatory graphic that is based on illustrations and diagrams. The answer, in my opinion, is that they are visual representations of data and phenomena that reveal things that the bare eye will not be able to see. The circles in the first example show a pattern that tells the viewer how the introduction of vaccines dramatically reduced the incidence of cases. The second allows the viewer to explore the structure of the cathedral, its building materials, the impact of the fire. They're both designed in a way the human brain can easily understand. The visual forms that are used to encode the data, the layout, the type, the user size, color, has been carefully crafted to make the viewer understand what the story is about. The challenge these two graphics address is that we're not good at processing large amounts of data, complexity, or huge amounts of numbers. We're actually wired to identify patterns. We are a very visual inclined species. That is why graphics, visuals in general, are so effective at communicating certain kinds of information. We, in the graphics team, try to take advantage of what the human brain is capable of and anticipate what our readers' brains are going to do with this information. Here's a very simple example of how this works on a very basic level. We have a sequence of random numbers in a grid. I'm going to give you 10 seconds to tell me how many zeros you can see. All right, time. So if you say 9, you are right. And I must say you have the eyes of a hawk. But for the rest of us, here's a little mind hack that could help. There you go. Try now. Just a slight change of color allows us to focus on the pattern of zeros almost instantly. Or even better, how is this? If we set back the noise of color by emphasizing even more this contrast, the elements we want the viewer to concentrate on shine like a Christmas tree. As I said, we normally have trouble processing complexity. Let me show you another quick example from my friend, Professor Alberto Cairo's book, The Functional Art. Imagine that you're working on the graphics on unemployment rates in the United States. And the goal is to display to your viewers what states in this specific moment in time have rates closer to the historical maximum. Would you represent the data as a table? Can you answer the question you're seeking to present by just presenting this table? So very quickly, tell me what 2 states have a current rate closer to the historical maximum. With that question, I'm forcing your brains to do something they are not naturally prepared to do. Scan the figures, read them, compare them, rank them. As I said, our job as designers is to anticipate what users will likely going to do with our graphic. What if we encode the data in a chart? Now we can easily see that Florida and California are actually the 2 states closer to the historical maximum. With just a gaze, this example illustrates one of the very principles of our work. If you have to design a visual piece, one of the first things you need to do is ask yourself, what is your graphic will be used to do? And what kind of questions will your graphic help to answer? And then use the best form of visual help the viewer, so the viewer can understand this information. Another principle we routinely apply is to establish a clear hierarchy in our graphics. There's an order in which you are going to scan with your eyes. Our vision tends to prioritize certain features before moving to other areas. We use that behavior to our advantage to craft a certain flow in the narrative of visuals. Take, for example, this graphic by Chris about breeding corals to make them more resistant to temperature changes in the environment. The graphic has a dominant visual element in the upper right corner, an illustration of the species of coral mentioned in the article. The viewer will most certainly start there before moving his attention to other sections of the graphic. Other chunks of information. The simple trick establishing a grid and hierarchy in our graphic allows us to direct how the information is going to be prioritized by the viewer. We also take advantage of some cultural conventions, like the West reading left to right and top to bottom, combined to reinforce this rhythm. Another design principle we like to apply to our production is a consistent use of color as a visual code. We often limit our palette to just 2 colors to limit the noise of contextual information by muting with gray anatomy portions, like in this graphic. The viewer can easily focus on information about the particles attacking the respiratory tract in red. Of course, the temptation of depicting reality as in a realistic way is always there. This extra layer of visual information don't really help our visualization to convey a clearer message. Here we have a figure from the author's paper on the left, and the same one redesigned by our team. As you can see, limiting the palette of the piece, the figure has now a lot less of contextual noise. Lastly, I would like to mention another concept we like to apply to our graphics, the data-ink ratio concept that was coined by Edward Tufte, the expert whose work has contributed significantly to designing effective graphic presentations. In his 1983 book, The Visual Display of Quantitative Data, he talks about the proportion of ink that is used to present actual usable data compared to the total amount of ink or pixels used in the entire display. The example on the left, based on this, we have a low data ratio. More ink is used for elements that don't encode data. The one on the left has a high data ink ratio. Less ink is used for elements that don't encode data. The idea is that graphics closer to a high data ink are more effective in communicating information to the viewer. Although there's no conclusive evidence that this hypothesis has merit, we like to keep Tufte's assertion in mind when designing our graphics. It's always a good exercise to try to clean any visual from unnecessary elements that are not contributing to the understanding of the data that you're trying to communicate. Another consideration is that a minimalistic approach often allows it to pack a lot more information in smaller spaces. Well, this is it basically. And I hope this brief tour into how our sausages are made at Science will help you in future visual projects, whatever they are. Thank you very much.
So our next presenter is Alice Kitterman. Alice, will you share your screen and take us to the next bit? Thank you all for posting questions in the chat box, and a couple of people into the QA. We'll come back to questions after the next, after the presentations are over.
Okay, hi everyone. I hope everyone's morning is going well. I am going to start my timer. As Alberto and Robert mentioned, my name is Alice, and I create the figures for the commentaries that are written by a third party about somebody else's research. And my main role is to create these figures for the sister journals, the online ones, signaling, translational medicine, immunology, and robotics. My role is unique in that I have to meet the needs of 5 different journals on 5 different publication schedules. So while I don't often have the time to develop the best graphic, what I can do is design a better graphic with a stronger narrative. So what I create is similar to something that I think that you all are familiar with, and that is a visual abstract. And these types of graphics serve a very different purpose from a research figure, and therefore should also look very different. So in this definition that I've put up here, the 2 words that I've highlighted in it are the key ones, and that is it has to be a summary and it has to be digested rapidly. So you may be wondering, where do you begin in creating this graphic? And you can look to the structure of a research paper. The typical story structure has many parallels to each of the sections of a research paper. And the conclusion discussion kind of serves as that overarching goal that answers the question, what problem is your research addressing? Telling this clear and concise story in a single graphic begins with the discipline to exclude information that shrouds that succinct summarization. Now, I like to think in flowcharts, as you see, and so I'm going to use this format to introduce some of the processes that I utilize to create my graphics. So the first thing that we want to do is to isolate the basic content of your graphic, and you can draw from your research paper. And then we're going to further curate or cull this information in the graphic. And finally, we'll design it to incorporate the visual breadcrumbs that help to promote effective visual processing. So again, to start, you can look at your research paper, and what you can start with is just to come up with one sentence for each of the sections. And among all these sentences, you want to make sure that somewhere you talk about the specific problem that your research addresses and what is the overarching goal. A bonus would be if you can actually try and keep this to 1 to 2 sentences. If you can combine sentences like material, methods, and the results, that would be great. And if you can achieve all these things, you can go to the next step, which is actually drawing out what's happening in each sentence that you've created. This is kind of an important aspect of it because it's starting to, kind of like get you into the visualization mode of your script. And finally, you want to take all your little pictures and you want to place them just in one particular page. And this the size is usually dictated, by the journal and the figure size restrictions. And once you've combined all of your pictures, you've basically created an illustrated version of your script. And now comes the hardest part, which is the streamlining. So some of the things that I start talking about when I'm looking at all of these different panels is I start asking myself some questions. And this is just an example of a few questions. For instance, you can look to see if all of your panels or some of your panels have certain consistencies in scale and magnification, in point of view, setting or background. And if they don't, this is where you have to start being very, I guess, analytical about creating all these images in your mind, with the same visual consistencies. If you've already got them, then this is where you can start trying to combine the panels. So let's go to an example. The way I've set up the examples are before, which is usually the reference material that someone like you all give to me, and then the after is what I produce. So what is the overarching goal in this before figure? It is just to show 2 different ways that potential antibody-dependent enhancement can develop. And what you see here is the left side of the screen kind of has this top to bottom visual presentation of the information. That's how you read it. And the right, you kind of start on the upper right side, and then you kind of swoosh over to the lower right side. So right there, you're already creating a visual interruption. And anytime you have this kind of visual interruption is an opportunity for you to lose the viewer's attention. You'll also notice that there are two kind of big cells. One is orange and one is gray, but they're the same cell. So what I've done on the right is I've changed the point of view for both of the images. And then I've also had this, kept this top to bottom kind of curvy, flowy like presentation of the visual information to unify these aspects of the figure. So this next example is a fantastic research paper figure on your left. Unfortunately, it's overly complex for a visual abstract. And so what I attempted to do here was just extract the important information in introducing this synthetic spider web and talking about like what it's made out of and how it specifically functions to mimic a natural spider web. So I'm trying to give the illusion of a single panel image by utilizing this ionic spider web as a backdrop and then putting the vignettes on top of it. And the vignettes describe, starting from the upper left side, what the fibers are made of, and then going clockwise around are all the ways that it functions like a natural spider web. Okay, and then so another thing that's somewhat similar is just trying to unify this background. So what is the overarching goal for this graphic on the left? The authors really wanted to emphasize the many different mechanisms by which calcium is moved inside, outside of a cell and around a cell. And so I think I've accomplished the same feat, showing all the different ways and mechanisms that calcium is moved in and around a cell, but I've unified it into one background, thus decreasing the number of visual interruptions. Okay, let's go back to the flow chart. So we've tried our best to eliminate multiple panels and images, but we still might have them, right? So if we still have them, we're going to, again, try to create an illusion, to reduce the number of panels that we see. So one of the things that we can do is, just is simply eliminating boxes and using blurbs as separators, or we can attempt to actually eliminate some panels. So in this example, on the left, the overarching goal of this figure was to introduce a new navigational controller that's added to this oldish quadrupedal robot. And what this navigational controller does is it now allows this robot to function autonomously. So in the left side, the top three images show different learning pedagogies that go into the design of this navigational controller. But these are kind of abstract concepts. So, and what I really wanted to focus on was what this navigational controller, allows this robot to do, which is navigate autonomously. So I eliminated the top three figures in favor of text boxes on the right. So the information is still there. And then this way, it kind of gives the illusion that there's like 4 panels. And it also allows the viewer to focus on what the navigational controller does. So are there repetitions in your figure? Sometimes repetitions can be a bad thing and a good thing. So let's cover the bad. The overarching goal in this before figure is to do a compare and contrast. So those are very important types of figures to show. However, because there's so many different elements included on this figure, all the different elements have to be reduced to a certain size, and that makes it harder to read from a visual standpoint. So what I did on the right is I tried to take that common element of the cells and the glucose uptake, the one thing that doesn't change, and I put them in the middle of the figure so that both panels could share, that specific element. And what I did was twofold, actually. One, I was able to create, to make all of the elements slightly larger on the page. And I'm also giving kind of the illusion that this is just a one panel figure. repetition can be a good thing. So the overarching goal in this awesome before sketch is that this particular robot is applying different learning strategies and actually affecting different outcomes. But at first glance, I thought, well, you know, is the robot thinking about two different things and choosing one, and then that's the outcome? Or is it thinking about two different things and it has the same outcome? So I thought in this case, it was better just to separate out the panels so that I could take the take home message would be that each different learning strategy that the robot uses affects a different outcome. Okay, so, you know, we're doing our best to eliminate these multiple images and panels to reduce the number of visual interruptions. But, you know, again, sometimes you can't. And so if you have to include multiple images, can you categorize them? And if you can't, you know, maybe they just need to be eliminated for the purposes of your visual abstract. Or if they can, then maybe you can group them. So in the left-hand panel, this is a great visual. The overarching goal is to show the evolution of deep sea exploration vehicles that are now bio-inspired. And so on the right, what I've done is I've used a grid pattern to organize the information so that it flows more effectively. So we've gotten through the painful part of streamlining, and now we're going to get to adding those visual breadcrumbs or those visual cues that'll reinforce that visual hierarchy. You know, Alberto talked about this. And these visual hierarchies, we're just going to use these visual cues to help tell the viewer, just directly tell the viewer, you know, what's the most important and what are the less important elements. So the first visual cue that I want to go through is size, which Alberto already touched upon as well. And it's also, you know, somewhat of a no-brainer. And so again, we'll go back to our quadrupedal robot. In the left-hand side, all the 7 panels have the same size. And on the right side, you know, clearly I've increased the size of the robot, you know, to show off that navigational system. Okay, and Alberto also touched upon this, color, and color is a very controversial topic. We've been taught since we were little that color should be used for description, the sky is blue, the sun is yellow, etc. But the unique thing that science does, and science graphics do, are that we use color functionally and not for description. So in this example, I hope this makes this clear. When you look on the left-hand side, where does your eye go? And what's the most important part of this graphic? So it's kind of hard to tell. So on the right, what I've done is that I've used a saturated warm color of red to highlight the receptor tyrosine kinase enzyme, which is the star of the show. And all of the other elements are still represented. They just are set back with the absence of color. Again, on the left here, what's the most important element or elements in this graphic? You know, I mean, you know, to me, immediately my eye goes to the left-hand side, you know, the full color panels, when in fact, all 6 of these panels are of equal importance. So on the right, I've used color specifically to draw your attention to those anatomic locations that are being highlighted. And on top of that, I eliminated the middle figure, which was unnecessary, which then allowed me to make all the individual panels larger. Okay, so this is an artist's secret weapon. You know, we have the ability to render very differently. We can use 3D, we can use 2D, we can use black and white versus form. So I think in this example is pretty clear where I want your eye to go, and that's to the squid robot and the squid. Organization is a graphic designer's secret weapon, as well as an illustrator's secret weapon, and it can make or break your figure. You want to use these layout and design principles to lead the viewer through your graphic. So in the left-hand side, this is a top to bottom representation. It's perfectly legitimate, but where it starts to fall apart is on the right-hand side with the the wedge graphs. And this is because vertical text is very difficult to read. So fortunately, this was a very simple solution. I just had to rotate the graphic counterclockwise, and that allows me to show the graph at the bottom to read it from left to right, which is much easier. And you'll also notice that the type of graph changed. And finally, detail. And, like size, this seems very obvious. And detail can mean adding more detail to a visual, or it can also mean just adding visual detail in general. So again, this example is probably a no-brainer. On the left, we have a flow chart, my favorite. But, looking at the flow chart, it might not be very visually interesting. And on top of that, you don't really get a sense of what it is unless you read it. So on the right, I've added that visual detail, which hopefully generates more visual interest, as well as adding those icons that might give a clue as to what this flow chart is about. So finally, it's a pop quiz. And I'm going to give you 10 seconds like Alberto did. Of all these visual breadcrumbs that I mentioned, color, size, treatment, detail, and organization, I want you to name 3 different visual breadcrumbs that you can add to make this more visually legible. Okay. Here's one solution. So I've done some color coding. I've also used color just for descriptive purposes while I'm setting other visual elements back. And I've used a grid organization, to organize the information. Okay, so, my hope in this talk was to really clarify why we creative professionals make all the changes that we make to your figures. And the takeaway message is really to give you a better understanding of our mindset and processes, so that in the future, the before figures that you give to someone like me will result in an after figure that was presented here. So thanks very much. And stop sharing. Okay.
Hi, as I mentioned in the chat, we're going to take a short 5-minute biology break before the next presentations. So we will resume in 5 minutes. Don't go away or too far. Hello, welcome back to Visualizing Science. We're going to continue with our program with Chris Bechtel. Chris, would you like to share your screen?
I'll share my screen and get going.
Okay.
Let's see here. Oops. Okay, good morning, everybody. My name is Chris Bickel. I'm Senior Scientific Illustrator at Science Magazine. I have been at the journal for about 15 years. I have a degree in medical illustration from the Rochester Institute of Technology in 2004, though I, as my title states, am distinctly a scientific illustrator at this point. I'm going to talk about editorial illustration at the magazine today. the bulk of what I'll discuss is cover illustration. That's the largest internal use of editorial illustration at the journal. Editorial illustration is artwork that's inspired by written text that visually illuminates concepts of a story. Not even necessarily the entire story, but enough to educate, while at the same time intriguing an audience and wanting to know more, wanting to read into the journal, wanting to get inside. These kind of illustrations can be whimsical and fun in nature. I think it's common these days to kind of see blocky editorial illustrations that are used in a variety of different publications. Within scientific illustration, it's still something that needs to educate and not purely be at my whim and fancy. So like I said, we're going to discuss cover illustration. Considerations that I use when designing cover illustrations are, of course, our audience. Our audience at Science is rather high-level functioning scientific researchers, but not everybody is an expert in everything. So we need to shape cover stories to be easily accessible for quick understanding and digestion of what the topic is, while at the same time even forming some questions to get someone to get inside of the magazine. One of the things that I consider is to look outside of just the scientific details for inspiration and how I might visually communicate a topic on a science cover. I try to leverage the freedom and leverage the opposite really of doing a didactic figure where we limit color. We try to use color a little bit more expressively on cover images, and then above all else, creating something that's beautiful but certainly accurate. There's really no point to beautiful scientific illustrations that don't tell a story. Here are 10 covers that I've worked on in the last couple of years, a little smattering of some of the ones I like. We are going to discuss the middle top 3 in more detail and look at how they are produced, and I will discuss some key considerations for each of them. As you can see here, we illustrate a wide range of science, of course. At the journal, we have perovskite solar cells in the bottom right. We have metamaterial cover, a surprising twist. We have a subatomic particles, molecular biology, chemistry, just a whole host of topics. So these are the 3 covers. Coronavirus Interrupted, that we will discuss about. That's a cell molecular biology piece. The central image, metallic solution, was a very interesting paper about an engineered fluid that turns into a metal while retaining the qualities of a fluid, or retaining the qualities of both, actually. And lastly, a surprising twist, which was a cover on Cheryl metamaterials and how they deal with compressive forces. So Coronavirus Interrupted was a project we worked on this year, of course. The paper was on the drug interaction with a protease and how the drug would potentially inhibit the protease and thus stop viral replication within a cell. In this case, be it that the story was about a small molecule drug here in orange and the beige protease, we needed to have a fair amount of context so that you weren't just looking at what would look like a random just protein database molecular coordinate illustration, and needed to add the context of coronavirus. And much of the work previous to this piece, a lot of the viruses I had looked at portrayed were in warmer tones. And I thought in this instance, it would be kind of fun to break that mold and also allow the point of the story and the warmer tones to really show in the forefront, while also adding the context needed to make the entire cover descriptive visual. So to break down how I started this process of making this cover, here's some sketches. At this process, I'm looking at compositional considerations for a final, how it might interact with cover logo, doing research and talking with researchers and getting boned up on the science, as well as getting my ideas down on paper, or in this case, an iPad Pro. Fun fact, I haven't drawn on a piece of paper in about 6 years. in this case also, there was a question while making this illustration as to whether or not the virus was a direct fusing virus or one that was taken up by endocytosis. And in June, we decided that the endocytosis model would be the best moving forward with our cover. So I graduated more to the right-hand sketch than the left. Next step is doing some preliminary 3D modeling to build base models, to research molecular coordinate data, and start visualizing some of my ideas in 3D space. Here I have designed on the right a full viral particle with M proteins, E proteins, and spike proteins, and then the viral membrane. I started working on with the left-hand sketch, trying to also kind of see if that direct budding concept would work. The sharp coronavirus-minded people will notice that the end capsids are all completely incorrect in the left image, and that was really just some preliminary work. Here, I use Cinema 4D, which is a Maxon product. It's a German 3D modeling animation program to set up my work. This is just a neat shot of kind of how that cover is visualized in the 3D space, how you see it before it is rendered out. And those on the webinar that do this sort of work will certainly know that the computer does not show you what you always get out of it. And it's your job to kind of understand that process and make the computer do what you want it to do. The next part of this process is rendering out individual components and elements out of the three-dimensional composition. This allows for rendering separate passes for isolating components and applying post effects more selectively than on the image as a whole. On the right-hand side, you can see as the image is composited together, my art director and I work together to start cropping this image to give it a little bit more of a focus visually and to get rid of some of the extraneous space that wasn't helping shape the story. So we're back to the final here. This sorry for being redundant, but this shows you our final illustration. In this instance, you may see that some of the orange, the bright orange drug molecules have been moved around so that the headline, deck and cover line can have good so they can be read properly and not be impacted by some of the visual components. That's a point that I will also touch on in subsequent covers here. This metallic solution was an interesting and far different editorial sort of take on a subject than the last example. In this science, an electrolyte transitions from being a fluid to being a metallic fluid, where you would have the complexity of having the dynamics of a fluid, but also with the characteristics of a metal. In this instance, I looked for some external cues. I looked to using, let me skip here, using water faucet imagery to give kind of a jumping off point to showing this liquid in a clean, clear, and concise way. Here are some sketches where I started sketching out some concepts. I had this initial understanding that this metal liquid kind of transitioned in a very finite and specific time point. You can see in the left side, I had sketched some fluids kind of falling off of a ledge, kind of playing up on a motif that this fluid hit a point of no return, and that ended up being incorrect. So I moved away from that to something that was just kind of inspired by the faucet imagery that I just showed. So the image on the right is where we kind of took this illustration. So at this point, using the water faucet imagery, I started hand sculpting this piece in Cinema 4D. I'm sure some of you have heard of ZBrush, which is a 3D sculpting program. I tend to do my sculpting in Cinema 4D. So this was sculpted a couple of times. I don't think that you ever need to go with your first attempt. It's really good to build off of your previous work and keep pushing where you're going to go with something. The two central images here are weren't right hitting the mark, but they were good kind of preliminary test runs to the image on the right, which is our final. The model was hand sculpted with a mouse, and I did some particle simulations to get some of the additional bubbling in the fluid and the spatters on the around the exterior. Here's our final illustration. It's very clean, very clear, kind of a high contrast look. The metallic solution cover line fits very nicely in that subtle curve in the fluid dripping, and that leads to just a nice continuity between the two elements and flow. We also needed to keep in mind how the teaser at the very top would be legible, and it's the bane of the illustrator's existence, all this type on these covers, but it's very important whenever designing these kind of pieces to ensure the whole package works clean and beautifully as a whole. The last cover I will talk about briefly was one I produced in 2007. The research is about very small metamaterials that, when compressed, do not squish out or bulge, but they twist and do not change in overall volume, not volume, but overall shape, like you would assume. This was a more explicit take on the reference material. Here's a couple of figures from the research. And in looking at these, I kept thinking of skyscrapers and kept thinking of kind of like old technical drawings. And I looked towards that sort of imagery for inspiration. Here are a couple of images that I used, and on the right was an initial kind of model that is actually twisting the wrong direction, but trying to, again, work with the first go and see how I could make this otherwise, I don't want to say boring, but square shape, you know, have some dynamicism and just some unique dynamic angular cues. And this, in the final, you can see that I colored the blocky shape with a gradient, which also helped give some verticality to the design. And I overlaid a few different line renders to try to get the kind of technical non-organic side of this across, while at the same time grounding the subject on the cover. This was a case of playing with cover with the subject coming over the science logo, which also added a nice sense of depth and some additional dynamic looks. So that is what I have for you today. I just want to give a brief overview of us using editorial illustration at the journal, some covers, and thank you very much. I'll turn it over to Scott and Nicoletta.
Thank you, Chris. So Niki, would you like to share your screen?
Okay. Hello, everybody, and welcome to my presentation on visualizing Alzheimer's with Dr. Scott Small. It's not news that artists and physicians have always shared a close working relationship. They need each other to make sense of intricate and mysterious workings of our bodies. Often physicians were also artists, or artists who also practiced medicine. Icons such as Leonardo da Vinci, Andreas Vesalius, and Frank Netter. Hi, I'm Niki, and no, I'm not a physician, although I did take a year of pre-med in college and have dissected a few bodies. Instead, I studied art, and the good news is that I'm a much better artist than I would have been a doctor. Here are some examples of illustrations I've done for Columbia physicians. Oops, sorry. Recently, one of our top caliber physicians reached out to me with my favorite question. I need art, can you help me? He's joining us here today. Welcome, Dr. Scott Small, the director of Alzheimer's Disease Research Center at Columbia, where he is the Boris and Rose Katz Professor of Neurology. Scott, can you please tell us a little bit about your research work?
Yeah, thanks to everyone for inviting me to participate in what I think is super important. The first thing I'll say that I am a physician, but unlike Da Vinci or Netter, I am not an artist. And that's why we need to learn to collaborate. And my prime focus is on Alzheimer's disease. I'll just make one general comment and let Niki take it from there. And that is, it's always surprising to me that sometimes scientists don't completely understand the value of art or visual representation. There's a sense, perhaps a hubristic sense, that science is science and visualization is just advertising, and we don't need advertising if our product is perfect. And that, of course, is wrong. Explicitly and implicitly. That's been proven wrong by practitioners of science, wrong by philosophers of science. I'll quote one or paraphrase one practitioner, and that's Einstein, who was very clear on you don't understand something unless you can visualize it. He did it in his mind. But of course, that's also true as we communicate science. The other point I'll make is that as science has gotten more and more complicated, what I was impressed by just watching our colleagues earlier in the earlier presentations is that particularly when you're going to high impact journals, and we all have a lot of, our science are like stuffed turkeys. There's a lot of things going on. Even scientist to scientist, it's really important to have that really great graphic to help communicate what we're doing. So no longer can you just show a bar graph and expect people to really read through the details. Often there's a panel that just helps explain the logic of the experiment, the experimental design. And so I do think that working with our visual artists on this call is super important.
Thank you, Scott. That was very eloquent. Thank you. Scott, so Scott was having his work published in Science Translational Medicine, and he was asked to submit cover art. He already had a few conceptual ideas that he presented to me involving the brain, tau proteins, neurons, clouds, tears, in Alzheimer's patients. So I began doing some rough conceptuals showing the neurons, the brain, tau, tears. It so happens that I have a 95-year-old mother who has dementia, so I ended up sketching her. But the art wasn't hitting the mark. The message wasn't quite clear. Basically, the problem with my approach was that it was too literal and not conceptual enough. Ultimately, Scott and I agreed that the cloudy brain raining tau was really the best conceptual idea to visualize. It told a clear story. It created a scientific allegory. Here are two versions using that concept. So Translational Science magazine used it for the cover in their November issue. Scott and I also worked on a series of black and white line images for his forthcoming book, Forgetting: The Benefits of Not Remembering. Scott, could you talk a little bit about that title? The title is really intriguing. Do you mind talking?
Yeah, it's a sly title, and it's a sly book for someone who focuses on trying to cure the suffering of forgetting. Mm-hmm. And so this really comes from the mirror image of that, from the emerging science of actually why the forgetting we're all born with. So everyone here on this call has forgetting, and I'll assume that no one here on this call has pathological forgetting, and we're all frustrated by it. But it turns out that Mother Nature in her genius has bestowed us with forgetting for good reason. And that's the essence of the book.
Scott, what is the most challenging part of working with an artist to realize your research?
Well, there was nothing challenging with you, Niki. So
well, thank you.
It's a bad example. But I think the hardest part we need to learn, the scientists. So in this book particularly, the hardest part for this book, this is meant for a general audience, will be published by Crown Random House, is really knowing how to elevate to a high enough altitude and get a bird's eye view on what captures the essence without being accused of dumbing things down. That is so hard for us. So that's what Niki and I have been working on.
Yeah. So in my experience, scientists work under tight deadlines, and their budgets are usually capped. They often don't have means or opportunity or time to engage with an artist. The main thing that drives scientists is the love of the discovery and sharing those discovery with people who can understand the intricacies. So for most scientists, art is an afterthought. The main goal of the scientists is to discover the knowledge. The dissemination is a second priority. Whereas the main goal of the artist is that of disseminating the concepts and ideas and narratives visually. It's important to bring art into the process early on. Many of the exciting discoveries are smaller steps along the way that are hard to explain, and visualization of data and mapping out research can lead to new hypotheses and insights for scientists themselves. Artwork can bring in additional funding. In other words, if the artwork enabled success at getting funds that wouldn't be obtained otherwise, the artwork would become the driver. Artwork needs to move from simply disseminating knowledge that is already codified to becoming part of the scientific process of model building. Finally, as with most things, the more attention and time given to its development, the better the results. So include art at the start. Also, I would like to, I am available to help anybody with their artwork in any kind of scientific capacity. And there is also a department, Columbia Creative, within the Office of Communications Public Affairs that is available to also help with artwork. So please feel free to reach out. And thank you for joining this webinar. Okay.
So thank you all presenters for that incredibly detailed and comprehensive overview and getting down into some of the points. We're going to take a few minutes and go through questions that were posted in the chat, and various panelists can respond to those. One of the overall questions that I think each of you could respond to are, what are the primary graphic tools or applications that you're using in your job? A bunch of people have posted, questions about 3D software and other detailed questions. So one by one, in the order that you presented, to start with Alberto, tell us what your primary tool is. What do you use the most, and what do you have sort of secondary in your toolkit that you find useful or helpful?
Adobe CC, the suite is kind of the base application that we use for everything, Illustrator, Photoshop, etc. On the 3D side of the equation, it varies. Like Chris and Bob, for example, use Cinema 4D, which is a standard in the industry in certain aspects. But Alice, for example, has used for years Blender, which is an open source 3D software that is extremely powerful and does the job. But on a very, if I have to answer this on a basic level, the primary tool that we use is the pencil. I mean,
Yes.
this is why we conceive everything. And in the case of Chris and myself, we use iPads since a long time, but it's the same principle. I mean, primary tool, pencil.
Alice.
Illustrator, Adobe Illustrator. I almost never go into Photoshop to paint. It's usually just for layering effects, and Blender. And then on the animation side, After Effects. But like Alberto said, Adobe CC has a lot of powerful, well, Adobe Fuse, I guess Adobe decided not to use that anymore, but that was kind of sad because it allowed you to create human figures and then and animate them a little bit so that you can freeze them in time and use those. And that was 3D. And Adobe Dimension as well, which is just great for smaller, less complicated 3D objects that you can then utilize in Blender or other 3D programs.
And what about you, Chris?
I'll echo largely that. We do use the Adobe suite as kind of the bread and butter, Adobe Photoshop. I use After Effects and Premiere for animation work. Photoshop is largely for color correction, some post effects. Really, bulk of my work is in Maxon Cinema 4D. I was exposed to that in college and have a hearty almost 20 years of use with that. So I have a particularly positive experience with it over the years. And also a host of small plugins and different smaller programs for visualizing proteins, getting three-dimensional coordinate data into the 3D space. And as Alberto said, as much as I use a digital pencil, like it's key to be able to draw, to get things down on paper or pad or whatever you have to do, be it digitally or traditionally.
And what about you, Niki?
Yeah, I would say the pencil is, yes, the humble pencil is the beginning of everything. But I too used Fuse. I used to use Fuse all the time. Before that I was using Poser, but both of those things are pretty much defunct. And Illustrator and Photoshop are my go-to, and then for animation, After Effects, of course. But nothing beats actually sketching out your ideas on paper first, and then going to generate computer graphics from that.
Yeah, sorry, Robert, I had something to add to that. I think Niki is completely correct in the fact that the computer doesn't make the art for us. It's a tool. It's another tool set, and your ability to harness it is up to your artistic skills and your brain and what have you. But at the end of the day, you've got to be able to draw. You've got to be able to visualize in a traditional sense and have all the skills that you would classically think an artist should have. And the computer, as much as we use it, it's not a substitute for skill.
Oh, sorry. Go ahead, Alberto. Go
No,
ahead.
sorry. Very, very briefly, and I would like to add that because I've seen questions about the three packages we use in specifically, but I would like to add that it does not matter what 3D package you use. They all have very similar functions, and they range in. I mean, the only difference, the basic difference is price. But as I said, you can use Blender, which is free source and is extremely popular and extremely powerful. But they have all the same functions and the same capabilities, essentially.
So I wanted to take us just a quick second and direct this same question to Dr. Small. Dr. Small, when you're working on a project and you go to talk to Niki the first time, do you do any drawing or visual conception, or is it all words? How do you approach conveying your idea for visuals to Niki?
Yeah, that's a good question. What I do, and for the scientists out there, it's the same way when you approach any expert, like a statistician, for example. first approach them with some general idea of what you have in mind. That's the best way we know epistemologically to communicate what we're thinking. The way I do it is, and the way I think Niki showed, and the way we did it together, is I admitted I can't use a pencil. So I went to the internet and simply found examples of what I had in mind just to get the ball rolling, or in this case, the pencil rolling.
Excellent. So I'm going to then turn that question to Niki and the other panelists. In your experience with scientists of all types, what would you suggest them to do to prepare? Is Dr. Small's experience of sort of pulling a few images from the internet the most helpful, or analogies helpful? What would you advise for scientists to do to help get the ball rolling?
It's very helpful. And one of the things that you have to keep in mind too is that an artist also has their own vision. So for example, I ended up going into representing my mother when really what I needed to do was something more conceptual and go back to the images that Scott had pulled out and found for me to explain what was really going on. And so it is really helpful to have the guidance of the scientists that you're working with, and the more detailed they can be, the better it is to be able to render something that's going to be useful to them in explaining their research.
Robert, could I make another point?
Sure.
And I don't know if there are junior colleagues on the phone, but there's a debate about how important it is to get a cover article. I think most people think it's a bonus. For those who don't know, and I know this with authority from talking to editors in the major journals, they really want a really striking cover. It helps them. They're not sure, even until a few weeks before publication date, on what they're going to decide on. And so don't be ashamed to ask them. You're actually helping them do their job if you think that you have a cover idea. And if they say maybe, try to generate something with the colleagues on this call or others, just to give them a sense. I think that really helps them when they make their final decision. Obviously, the quality of the science matters, if they think it's really a lead article or not. But there could be multiple options. They'll go after something that I think is visually pleasing. So this is a dialogue with the editors, even pre-pub.
So that's a jumping off point for a question that was posed in the chat. Selection of cover images, are there standards? And also, who decides on the cover layout, the text, what gets superimposed over what? Is that all in-house, or do external people contribute? At some point, there were other external contributors or individuals were suggesting artwork, and now it seems to be done in-house. So people want to know how this works.
The covers, I can talk about covers because we really process a lot of them, and my team is involved in a good portion of them. So we decide the covers in house normally, but again, the primary consideration is the material from the authors. The authors bring us their submission or their ideas for the cover, and we work as a group, and it's interdisciplinary actually. Several groups work on this idea and how we can develop. I have to say also that we try very, very hard to make our covers very consistent with our style and the style of the magazine in general. So there's a lot of debate in this cover, lots of back and forward. There are cases in which the submitted art from the author is goes as is sometimes, but there's also a dialogue, even in this cases, with them on how they can help us to make the the cover more close to our style and on our aesthetics in general. So it is a back and forward, but ultimately the decision is ours, and it is a decision shared by the design group, by the people who direct the editorial, and so it is a consensus, and it is a long process. Actually, it takes us a long time to decide covers and how we're gonna finally publish them.
Robert, I can speak to some of the more artistic, I think, points of that question. When I propose a cover, I tend to design it and present it to our designers, our creative director, who is the individual that has the skills and the decision to decide where the cover line is going to be, how everything's going to be assembled. And so I would say largely those kind of decisions are mostly done in-house. We like to have, I don't want to use the word control, but in getting art from other people, vendors, freelancers, et cetera, or researchers, sometimes we don't have the flexibility to make it look how it needs to look from a professional, quality standpoint, as well as just making a really good, engaging cover. So I'd say most of the time we use submitted work as a jumping off point, but we work, like Alberto said, in direct concert with the researchers and our editorial team to decide what's really going to be the cover of Science. I think I would venture that the science drives it more for our journal than, so I mean, if we have a really compelling research that needs to get on the cover, but not good imagery, we will make that happen. So I would say the art does not drive it terribly deeply as much as the content.
So it's more of an editorial driven choice.
It is initially, and then when we get deeper into it, the aesthetic and our opinions start to matter more and more and more, and we shape that narrative and that visual as we get to that point.
I understand this is a highly competitive. Everyone wants the cover. Everyone feels their work should be on the cover, or they wouldn't be proposing it.
Yes, and that is and that brings me to another thought process that I was thinking and hearing you ask the questions, is that sometimes in the scientific community, people are so wed to their ideas and they're so unbelievably good at what they do that they have a hard time having a professional visualizer come into the fold and want to change a couple things or listen. So I would say, as a general comment, embracing us. We're around. Not every researcher is going to have the budget or time to get a top level illustrator, and that's understandable. But look and embrace those other professions and the people that can help shape your work.
And I may add that in many times, the author seeks our assistant actually to develop concepts for the cover.
Yes.
And I mean, just to kind of like, go way back, to, what a scientist, how a scientist should communicate, with a creative professional like us, is really just to go back and kind of, present a narrative to us, tell us a story of, what is the big picture, and how that research fits into the big picture. And, we do, a lot of us do have science backgrounds, or we go through a lot of science study. And so we're not experts, and we are relying on you to tell us, but we do have, some training, that helps us bridge that gap between the science and the visualization.
Great. Alice, while I've got you on a roll here, there have been a couple of comments or questions about color palette, font selection, and all those types of things. Can you shed some light on that for our audience?
Well, again, the great thing about what Science has done is that it's tried to, we've tried to create a strong visual brand that is applied across all of the journals. So when we talk about, the use of color, as a functional versus description, we're really creating those visual hierarchies that basically tell the viewer what to look at, what's the most important element or important elements, or what is relative to each other, what relates to each other. You can use a family of reds, to denote a protein family. We start with a set of guidelines, font sizes, font weight, line weights, leader lines. And that gives kind of like that visual continuity across all the visuals, fewer visual disruptions, which is what we're talking about. So, someone did ask about the color. We are very mindful, of colorblind, our colorblind audience. So we avoid red, green where possible. However, it's not really the red or the green, it's the value. It's the saturation of those colors that can also make that distinction. So, we use those tools, to show us, if the values are different enough. And that's why, often you'll see the teal and orange juxtaposition, in some of our palettes. So we do start with that set of guidelines. And then, we work with the authors and researchers. If they tell us that, a specific color needs to be used, for specific elements, if they can justify it, sure, we'll go with that. Oh.
Yeah.
Oh, you're muted, Robert.
Yeah.
I wanted to wrap up in just a minute and sort of circle back to this notion of dialogue between the researchers, scientists, and artists. Maybe Scott and Niki could talk a little bit about their day-to-day interaction. Scott, what guidance are you giving to Niki? Niki, what guidance are you giving to Scott? And how do you reach a point where you're both satisfied with outcomes?
Well, Scott is good at working with artists to begin with. He has an open mind, so he's pretty good at being okay with thinking out of the box, so to speak. But sometimes that's a little bit of a problem, since scientists are very much in their work and they kind of have a tunnel vision. And so sometimes it's a little hard for them to see out of that, to make that leap.
So I'd like to maybe answer with a meta point on maybe the inverse of what I said earlier, to compel us, the scientists, to reach out to you. I don't know if this is an uptown or downtown problem in our beloved campus, but I can tell you that I know a lot of my colleagues uptown would love to have better art rendering, and they simply don't know the facilities and the colleagues we have here on the phone who can help us with that. So
they're welcome to come to me.
No, I know, but insofar that this could be communicated at every level, I think the spreading the news is important.
Yes. Mm-hmm.
And I think it's important to acknowledge too, maybe it's a numbers problem. I think there's a lot more researchers and scientists in the world than scientific illustrators. So that's a bit unfortunate. But I think that's a job that, our community needs to do, should do, and maybe can do better about, like groups like the GNSI, the AMI, professional organizations try to get, that message out. But we are kind of a niche group, and it's a unique speciality.
By the way, Chris, I just wanted to say that your twist cover was very much admired by one of our professors at Columbia. So a while ago, and he brought it to me and he said, "Look how great this is." So I had it up in my office for a while. I didn't know it was you that had done that, but now I meet you, so pretty cool.
Well, we're at our 11:30 allotted time. I wanted to thank all of our presenters and the organizers of this event, and particularly Niki Barolini, for putting these experts together. We'll continue with this series. There are 2 more sessions of Visualizing Science in the coming weeks. We will be promoting those widely and try to get as large an audience as possible. Thank you all and have a great day.
Thank you.
Thank you.
Thank you.